Okay, so let's look at what a myosin molecule really looks like at a close-up of it, and then we'll look at actin. Myosin actually looks like two golf clubs that are twisted against each other. Okay? Two long rods twisted. And they got two globular heads at the end. But we have a bunch of these all tacked together, and this is what your myosin actually looks like. If I went back to this one here, see all those little bumps on this blue over here? That's all the myosin heads. We're just getting a really close-up of it. Now, each globular head has two binding sites, one for ATP and one for actin. Okay? Now, we got to put it together. You understand the anatomy of a myosin over here? We got to show you how it's going to bridge with actin. So let's talk about the anatomy of actin first. Basically, what it is looks like an actin molecule is two string of pearls twisted with each other. That's the purple here. And each one of these molecules, these purple beads, has a myosin binding site there. You can see the darker end of it. But it's being blocked by this orange stripe here. Alright, so it has that myosin binding head, which isn't, that's what it is right there, though. So right now, if myosin is underneath here, it can't bind to that site because something's blocking it. This orange thing that's called tropomyosin. Tropomyosin is blocking that spot. This tropomyosin is attached to this yellow thing called troponin. Troponin has a calcium site on it that calcium must bind to. When calcium binds to the troponin over here on the yellow, it changes shape and allows the orange band over here to move over, exposing the myosin binding site. Now myosin can bind on here and move it. Calcium is needed to expose that myosin binding site. Okay, so it's trying to show you over here, the exposed sites you can't see here. But once we have calcium over here, it's going to go onto the troponin over here, move it out of the way, and now you got the myosin binding site exposed. And the myosin can bind to this. Okay? Troponin is not shown on this, but when the myosin binding sites are exposed, the myosin heads can bind over here and kick back and cause a contraction. As long as those myosin binding sites are available or exposed, you will have contraction. You need calcium to do that. So where's this calcium coming from? All right, I'm glad you asked. Let's go into another topic. We have these transverse tubules, also known as T-tubules, and they're an extension of the sarcolemma, the cell membrane. And they extend into the muscle itself. And they ride right against the sarcoplasmic reticulum that contains calcium. So that's where the calcium is coming from. It's coming from inside the sarcoplasmic reticulum. The sarcoplasmic reticulum, when it comes very close to the T-tubules, and I have pictures to show you this, it's called the terminal cisterna. It's the part of the sarcoplasmic reticulum that is close to the T-tubules. And the triad is when we have two terminal cisterna and one transverse tubule. So let me show you what we got here. We have a neuron that's going to be attached to a muscle. Muscle. This electrical impulse that goes from the neuron to the muscle goes down the axon. This electrical impulse is called an action potential. This action potential is going to go down this axon, and it needs to stimulate this muscle to contract. Where the neuron touches the muscle, we call it the neuromuscular junction. We'll save that for tomorrow. But when it goes across there, then it's going to send its action potential onto the muscle, onto the onto the sarcolemma, the cell membrane of the cell. The cell membrane, so this is the sarcolemma, and it goes in. It invaginates into the muscle itself. When it does that, we call these T-tubules. They look like T's. And the way this is set up, I'll show you the video of this so you can see it. But there is sarcoplasmic reticulum all in here. Underneath the sarcoplasmic reticulum, you will have all those sarcomeres, all the myofilaments. So they're underneath all here. And the sarcoplasmic reticulum is loaded with calcium. The blue is all sarcoplasmic reticulum. When it comes close in contact with the T-tubules, so this is the T-tubule, this is the terminal cisterna. Terminal cisterna. Terminal cisterna is the part of sarcoplasmic reticulum that is adjacent to the T-tubule. When you have this area here, a terminal cisterna, a T-tubule, a terminal cisterna, when you have these three things together, we call this a triad. So good? This is what I've been trying to draw. Sarcoplasmic reticulum. The yellow here is the T-tubules. Alright, we get up closer. See it? T-tubules, sarcoplasmic reticulum. The area of the sarcoplasmic reticulum that's adjacent to the T-tubules, these areas are the terminal cisterna. You put the three together, you got the triad. You get really close up, and that's what I'm trying to draw there. The sarcolemma is up here, and when it invaginates into the cell, this is called a T-tubule. sarcoplasmic reticulum is all here in the blue. When it's found adjacent to either side of the T-tubules, we call that the terminal cisterna. These three areas here, terminal cisterna, T-tubule, terminal cisterna, we call it the triad. Underneath all the sarcoplasmic reticulum is our myofibrils, the actin and myosin. So what will happen, this action potential that the nerve transferred onto the muscle fiber is now going to send the action potential down, because it's all connected, the sarcolemma and the T-tubules, it's the same stuff. It's going to go down into the T-tubules. It's electrical impulse. And somehow, we don't understand the precise mechanism, but it actually tells the terminal cisterna to release calcium. And now the calcium is going to flood all those actin and myosins. And like I showed you before, calcium is then going to go to troponin, going to move the tropomyosin out of the way so that it can actually expose the myosin binding sites on the actin, so that myosin can then bind to the actin.
We move our bodies by shortening or contracting our muscles. Let's take a closer look. A muscle consists of parallel muscle fibers. Each fiber is a single cell in close contact with a motor neuron. Each muscle cell contains bundles of parallel myofibrils, shown in red, surrounded by endoplasmic reticulum, or ER. An action potential traveling down a motor neuron initiates an action potential in the muscle cell. The action potential spreads along the membrane and down tubules that extend into the cytoplasm, causing the myofibrils to contract. Each myofibril consists of a series of sarcomeres arranged end to end. The sarcomere is the contractile unit of muscle. Each sarcomere consists of thick filaments of myosin, shown in purple, and thin filaments of actin, shown in orange. A sarcomere contracts when its actin filaments slide past its myosin filaments. Contraction shortens the sarcomere, but does not change the length of the actin or myosin filaments. The myosin filaments have heads that bind and pull actin repeatedly, shortening the sarcomere. How do the myosin heads pull actin? In a relaxed muscle, the myosin binding sites on actin are blocked by a protein complex, and ADP and phosphate are bound to each myosin head. An action potential causes the ER to release calcium ions, which bind to the protein complex, causing it to shift and expose the myosin binding sites. The myosin head binds to actin. When ADP and phosphate are released, the myosin head bends, pulling the actin. This is called the power stroke. Next, ATP binds to the myosin head, causing it to detach from actin. When the ATP is broken down to ADP and phosphate, the myosin head extends. The sequence repeats as long as calcium ions are present. Bind, power stroke, detach, extend. The combined work of many myosin heads causes the actin filaments to slide past the myosin filaments. When the action potentials stop, calcium ions are pumped back into the ER. The myosin binding sites on actin are again blocked, and the muscle relaxes. During muscle contraction, actin filaments are pulled toward the center of the sarcomere. The simultaneous contraction of the sarcomeres shortens the myofibrils, and the entire muscle cell contracts. This is how a muscle cell responds to action potentials in the motor neuron. When many muscle cells contract together, the result is movement.
So you have this muscle contraction that occurs. Now, if the myofilaments are going to contract, well, that's just going to make the organelles contract, but not the whole cell. So the myofilaments have to be connected to the sarcolemma. It has to be connected to the cell membrane. Because once the cell, does that make sense? If this contracts, it's got to connect. These things have to be connected to the cell membrane. Otherwise, the whole cell is not going to contract, just the things inside. Does that make sense? So we have to have something where it's going to be connected. So we have all of this that's going to contract, but then we have a molecule in red here that's called dystrophin. That's going to connect the sarcomere with the cell membrane. There's our phospholipid bilayer. That's our sarcolemma. So when this contracts, and you have another one that's going to connect over here, then the whole cell's going to contract with it. Does that make sense? Can you see if we miss, if let's say dystrophin is not there, what is that going to do? If you don't have dystrophin here, yeah, what's not going to contract? No, these will contract. This won't contract. Does that make sense? This will still contract. It's like taking a balloon and, I don't know, if something inside is going to contract, it's not going to make the balloon contract unless that piece is glued to the balloon itself. Then it's going to even make the balloon itself contract. But you could have something loose in there, it's not going to do anything to the cell itself. It's just inside will get contracted, but not the outside. Does that make sense? So you need to have this so that the whole muscle will contract too, not just the myofilaments. So dystrophin has to be in there. It's a protein that's going to link the actin and the endomysium over here. Okay? And it transfers the force that was in here to the cell membrane. You've got to have that. Okay? So it reinforces it. Well, we have a disease where you don't have dystrophin. What's that disease called?
Muscular dystrophy.
That's muscular dystrophy. Muscular dystrophy is where you have a decrease in the amount or just not working of the dystrophin. All right? If you know about the Labor Day telethon that comes out each month or each September, Jerry Lewis, I don't know if you guys know who he is, but he's the one that started it, and it's about muscular dystrophy to raise money for that. It's an X-linked disease, which means, is it equal where females and males get it? Is it more males and more females? X-linked. More males have it, a lot more males. But that's what that is. That's a genetic disease, okay? And what happens here is that there's a mutation in the gene that makes dystrophin, which means that the myofilaments contract, but the cell doesn't. So therefore, they're not going to be able to contract things. If you need to move things, contract, like lift things up, okay? There's something called Gower's sign that we see in childhood. I'll show you a picture of it. But basically, it's for them to get up, they need to, see what happens with muscular dystrophy, their thigh muscles get affected first, and then it moves downward, then the lower legs, okay? So what happens here is that if a child, let's say a 5-year-old boy or a 4-year-old boy, is on the ground and they need to get up, they'll walk themselves up with their hands because their thighs are very weak. So they're going to use their arm muscles to lift themselves up, and then their calf muscles will lift them up that way. But they can't really use their thigh muscles to do that because they're so weak. And it's called Gower's sign. I'll show you what that looks like. Okay? There's two types, and it's death usually when the respiratory muscles don't work, like the diaphragm. Okay? There's two types of muscular dystrophies. There's something called Becker's and Duchenne's. Duchenne's is more severe. They get death usually in their 20s. Becker's usually less severe, but they usually get death in the 50s or 60s. Again, the same reason is when the diaphragm doesn't contract properly. Okay? And that's Gower's sign. So when they're getting up, they're using their hands to get up, but that's not the only thing that you could recognize. you could look at the thigh muscles and notice how thin they are. Or look at the calf muscles and look how massive they are. So you put it all together, what's happening here? Because it could be just a little quirky thing the parents see. He likes to get up with his hands. But a keen doctor or a keen medical professional will see that the legs, where the muscle distribution is. Look how large those calves are compared to the thighs. You're putting everything together. Okay? So that's muscular dystrophy. Questions about that? Alright, the last thing I'll do very quickly is nerve-muscle relationships. Okay? Basically, nerve must stimulate a muscle. This kind of thing here. Alright? We have a 1-to-1 ratio. Each terminal end must go to one muscle fiber. Now you could have this going to another muscle fiber. We could have another one going over here to another muscle fiber, and another one over here to another muscle fiber. They won't go to a nerve. It'll either go to a muscle or go to a nerve. Does that make sense? So what it is that each terminal end needs to go to a muscle fiber. Okay? So a motor neuron is this. We're going to get into this much later on, but I want you to see it now. This is a cross-section of your spinal cord. We're going to have something going into it and something coming out of it. You touch something here like your skin, we're going to have sensory going into the spinal cord, and then the spinal cord, if it goes up to the brain to interpret what the heck that is, the brain is then going to send another neuron to go down, and this will be what we call a motor neuron. And it's connected to a muscle. So in other words, if someone touches you over here with a needle, then it goes up to the brain, your brain will sense that's pain, and your brain is going to say, oh, we're going to move our hand away. It's going to contract a certain muscle here. And that's what's going on here. All right, so you have the neuromuscular junction here where the neuron actually goes to the muscle here. Okay, so it carries that. If it goes to a skeletal muscle, we say it's a somatic motor neuron. If it goes to something inside, like a smooth muscle or gland, then we call it a visceral motor neuron. Okay, so a motor unit is this. All right, a motor unit, and a motor neuron, and all the muscle fibers it controls. So here's one neuron, but it can have a lot of terminal ends to different muscle fibers. That's a motor unit. One neuron and all the muscle fibers it controls. So like what I did on there, there's more terminal ends, but here's one motor neuron, and it actually will go to 1, 2, 3, 4 different muscle ends. Does that make sense? Muscle end plates, so to say. That's one motor unit. Okay? Cell bodies of motor neurons will be located in the brain or spinal cord, and they have long axons that go way out. So the cell bodies are here, but we have a long axon that goes that way. Neurons are very, very long. They're thin. You can't see them, but they are very long. All right? The axon terminals are basically where that end is of the axon. It has other names like synaptic bulbs, terminal buttons. They're all the same thing. You know how to say one structure about many different ways, right? So that's what happens with that, okay? And the motor end plate is the area of the muscle that terminal end is going to touch. They're not really touching, but we'll talk about that on tomorrow, okay? So the motor units, and there's two motor units in this picture here, one in blue and one in red. There's usually about thousands or hundreds and thousands of them going out here. They're only showing two. So what happens here is postural control. We have one, let's say the blue one is going to turn on a certain set of muscles and contract them, whereas the red ones are going to relax. So one contracts, one relaxes. They're still going to the same muscle, but different fibers. This allows the muscle not to tire out so fast, even though we're contracting it. Does that make sense? We're only going to be contracting certain muscle fibers, and then when that gets tired, the other ones will start doing it. But I only drew two, there's only two up here, but there's hundreds of them. So they're all doing it that way. and it allows the muscle not to fatigue. That's why I can stand up for 10 hours giving lectures, because my back doesn't tire out because I have so many different motor units going to my back. Okay? We have fine control, and we have small motor units that contain as few as 20 muscle fibers per nerve fiber. These tire out fast, but they move fast. The best way I can explain it is eye muscles. If I want to look straight, but I want to move my eyes to the right, I'll do that, and I can do it fast. In fact, I can make it go right and left very fast, back and forth, but it tires out very fast. Does that make sense? All right? So then that's because we have less motor units in our eye muscles. But we got more in our back, and that's why it doesn't tire out. But it doesn't. I can't move my back as fast as my eyeballs can shift back and forth. Okay? And then we have large motor units containing hundreds and thousands of muscle fibers per nerve fiber. That'll be like your gastrocnemius muscle, your calf muscle. I can lift that up with great strength to lift up things. Okay?